I'm just going to give a brief introduction to the, sorry, my Google is thinks I'm asking a question. Okay, I'm going to give a brief introduction to the project that that's put this workshop together, called Easy Rider, and then I'm going to hand over to Toka to lead the workshop today. So Easy Rider is a project funded by the UK government, and we're interested on bringing together expertise on automated computer vision systems for monitoring insects. And our interests are very broad, both spatially and technologically. And it's an all-inclusive networking project that puts together sessions like these. So we've got these 4 different strands of work that we're undertaking. Establishing community of practice, so trying to bring together people and projects that are involved in the automated monitoring of insects using cameras and computer vision technologies. Building opportunities for knowledge exchange between computer scientists, ecologists, citizen science project organizers, arranging field trials to test out systems and compare systems and setups across the world, and putting together the things that we learn into a best practice guide. This workshop is one of these knowledge exchange activities, and you'll hear more about additional workshops coming up in the future. We've got 2 more planned in the next couple of months, one focusing on machine learning and one focusing on comparing automated methods to traditional methods for surveying for insects. So look out for information on those. So without further ado, I'll hand over to Toka, who will introduce today's workshop.
Thank you very much, Tom, and thank you all for showing up for this event. We are really excited to have, like Tom say, people from all around the world join us today for a discussion about hardware design in particular, and particularly revolving around the hardware design of traps that can be used for monitoring nocturnal insects, in particular with non-invasive image-based methods. So you've probably all seen the list of speakers for today. We have both speakers from Australia and US in the same meeting. We're kind of proud of that. But we also have attendance from many different places, and we thought it would be good to spend a little bit of time going through a round of introduction, because we're also trying to grow a network around the world that can help make people aware of these new technologies and the pitfalls and advantages. So we have this one slide that we put together on essentially the expertise that you typed into the form for signing up for this meeting. Gives you a little bit of an impression of obviously the broad topics that people are interested in, but also some of the more marginal reasons for joining this event, and how we might be looking at other aspects in the future. But without further ado, I think we will be spending like the next 10 or so minutes going through a brief round of introductions. Basically, we want you to say your name and where you come from, and then we'll ask you to say a few words about your motivation for joining this workshop. So we'll try to capture that information as we continue this conversation. And I'll try to do this in the order that I see you on the screen, so there's no confusion about who is next. So if you haven't, please update your name so that I can introduce you by your name. I think most of them are easily readable. So name, affiliation, and motivation for joining this workshop, and try to be brief so we have time for the discussion afterwards as well. I'll start with you, David Goddard.
Good afternoon. I'm David Goddard. I'm an ecological consultant specialising in invertebrate surveys based in the Midlands within the UK. And I'm just beginning to get into using other technologies other than the standard survey methods of collecting specimens and IDing them. So one of my colleagues is also looking at bioacoustics.
Great. Then I've got Mark Bellal.
Hi, Mark Bellal from Sherbrooke University in Quebec, Canada. I'm part of the Easy Rider team, and I'm mostly interested in the output of these moth traps, so to monitor insect populations around here. Thank you.
Great, thanks. And Kim Biao?
Yes, I'm Kim Biao from the Aarhus University, and I'm working with electrical and computer engineering, and I'm a primary teacher, and I've been doing a lot of the developing of the Aarhus moth automated trap that I will speak about later today.
Great, thanks, Kim. And Laura Buffy?
Hi, I'm Laura from Italy, Rome, and I'm a designer. I work for the Joint Research Center of EU, and I'm interested in pollinators camera and monitoring for a project I'm doing.
Great, thanks, Laura. Simon Teagle?
Hi, Simon Teagle, UKCEH in Wallingford in the UK. I work closely with Tom and David Roy and Albert on moth monitoring and developing the moth trap that we originally got from Aarhus, Toka and Kim. And I'll be talking more about that later.
Great, thanks, Simon. And Joe Bowden?
Hi, my name is Joe Bowden. I'm a research scientist here in Corner Brook, Newfoundland with the Canadian Forest Service, Natural Resources Canada. A big part of my work that we do in this region is focused on understanding the impacts of eruptive forest insects in the forest, and also on non-target impacts of spray programs, for example, so macromoths. Cheers.
Great, thanks, Joe. Maximilian Sittinger?
Hi, my name is Maximilian. I'm a PhD student at the US Green Institute in Darmstadt, Germany, and I'm currently working on building and programming a camera trap for automated detection of flower visiting insects. With this, you want to have an alternative to yellow plant traps for insect monitoring.
Great.
Thanks.
Thanks very much. Gunnar Brehm?
Yeah, hello, I'm Gunnar Brehm from the University of Jena. I will give a talk later, so you will learn more about what we are doing here. But basically, I'm a moth ecologist and taxonomist.
Thank you. Jeremy Golnick?
Yes, I'm Jeremy Golnick. I'm program manager for wild pollinator conservation for a French NGO in France. And we are developing a participatory science program using a moth monitoring system.
Great, thank you. Donald Hoban?
I'm Donald Hoban. I'm based in Canberra, Australia, and I'm mostly doing this out of personal interest, but I want to increase the availability of time series data for invertebrates.
Great aim. Great, thanks, Donald. Luca Pecoaro?
Hi, I'm a postdoc at the WSL, which is a Federal Research Institute in Switzerland, part of the UKCEH. I'm working both on the hardware side of camera traps for pollinators monitoring, in the alpine especially, but also on the software side, developing models to detect and identify pollinators on flowers.
Great, thank you, Luca. Stefan Petersen?
Hi, I'm Stefan Petersen. I am the founder of FaunaBits, and we built the Diopsis camera, which is a yellow plate with a camera in front where you can detect most of the insects which are flying.
Great, thanks. David Roy?
Hi, I'm David Roy from the UK Centre for Ecology and Hydrology, working with my other colleagues on the call here. My main interest is in biodiversity monitoring, particularly Lepidoptera. So I'm interested in how insect camera traps can complement other approaches, particularly citizen science approaches. Thanks.
Thank you, David. Elke Jongjans?
Hi, yes, I'm Elke Jongjans from the Radboud University in Nijmegen in the Netherlands. And together with Stefan Petersen and other people, we're working on the Diopsis smart camera. So yeah, really interested to hear this and witness this workshop.
Great, thank you. Jamie?
Hi, I'm Jamie. I work for Aarhus University. I'm Toga's postdoc. I'm also affiliated with the UK Centre for Ecology and Hydrology. And with Toga, we've had some success monitoring moth pollination in the wild using time-lapse cameras. And I'm interested in learning about standards and best practice in handling entomological images.
Thank you, Jamie. Jacob Edek.
Hi, I'm Jacob Idek. I'm from the University of Florida, working with Rob Gorelnik. I'm a PhD student. We're especially motivated to gain some expertise in light trapping, learning from some of the folks in Europe and elsewhere to start building out a network of light traps in the US. We're also interested in like data standardization, especially.
Great, thanks. And Sine Brinkblue. Maybe she's not able to speak right now. I know she's traveling. Okay, Julian Evans.
Hi, I'm Julian. I'm with the University of Amsterdam, working as a data scientist on the ARISE project. And as part of that, we're interested in looking into a lot of different sensors, which includes the Diopsis camera, but we're also interested in looking into the our house moth trap.
Great, thanks. Jörg Freihoff.
Hello, I'm from Berlin in Germany, from the National History Museum, and we are setting up a long-term monitoring scheme for moths in urban environments, basically based on citizen science approaches, and I'm very curious what to learn from this meeting. Thank you.
Thank you. Neil Kopp.
I actually put my full introduction in the chat for reference, but I coordinate SCAN, which is a arthropod data portal focused on North America, and I deal with hundreds of collections. And so my primary role is to deploy models into the field and work with various collections and ecology programs in doing so.
Great, thank you, Neil, and thanks for putting it in the chat. Feel free, everyone, to do that, so we have a record of that if you want to. Rob Gorelnik.
Hi, everybody. So I'm from the University of Florida, where I'm a curator and professor. And I mostly work on biodiversity questions where I try to hybridize between ecological theory and data science approaches. And the key motivation for me in regards to developing sort of scaling up approaches for automating insect monitoring is we've been working on urbanization drivers on insect abundance and phenology. And so scaling up how we're automating light trapping and using citizen science approaches is critical for kind of being able to look at these questions more than just at the local scale or regional scale. Thanks.
Great, thanks. Johan Beckman.
Hello, I'm Johan Beckman. I work at the Lund University in Sweden at something called the Biology Technology Lab. So I work together with Lars Pettersson, who's running a national insect monitoring program. And he's been giving me the task to assemble and test some automated traps. So I've copied the design from our neighbor country, Denmark. And we're just during these days trying to get it running and test it.
Great, thanks. Katie August.
Hiya, I'm a PhD student from the University of Aberdeen in Scotland, and I'm just kind of starting my computer vision journey with the aim of trying to ID individual goshawks using camera trap images. So I'm kind of here just to see what else is going on with computer vision.
Great, thanks. And I hope this works now. Let's see, David Rolnick.
Hello, everyone. I'm David Rolnick, and I'm a professor of computer science at McGill University and the Mila Quebec AI Institute, where my group focuses on applications of machine learning and AI to problems relevant to climate change, including mitigation and adaptation, and a lot of work on biodiversity monitoring. I've also worked on moth monitoring and moth watching as an amateur for a long time, so I'm really excited for our work as part of this collaboration. We're trying to essentially develop a clearinghouse for machine learning methods that can work across lots of different traps, and developing a suite of tools that are essentially hardware agnostic. or can quickly adapt to individual hardware setups. So I'm very interested in talking with y'all about that. And we'll have a session on focus on the machine learning in a few iterations of the webinar in a couple of months.
Great, thanks, David. Daniel Kissling is next.
Yeah, hello everybody. I'm Daniel Kissling, University of Amsterdam. I'm mostly a biodiversity scientist, and we in the context of the ARISE project, we are currently setting up a few sites in the Netherlands to test various digital sensors, including acoustics and cameras. And we have one of the Aarhus moth cameras here that we wanted to deploy this year, and we have also a range of Diopsis cameras that we are setting up.
Great, thanks. Michael Bunsen.
Hi there. I'm Michael Bunsen. I'm based in Portland, Oregon, on the West Coast in the US. And Michael works too. But I'm primarily a developer and DevOps engineer. I've been working with the eButterfly project for the last couple of years, and more recently David Rolnick and Aditya. I am interested in implementation of research and primarily in applications and food systems and sustainable food systems and where they intersect with biodiversity and conservation. Thank you.
Well, great to have you. Oregon must be an awkward time zone right now.
Yes, thank you. I'm just getting started here.
Right. Aditya is next on my list.
Yeah. Hi, everyone. My name is Aditya Jain. I'm a graduate student at the University of Toronto and also an intern at Vila. So with David Rolnick, I'm building machine learning models for classifying, detecting, and tracking moths in the trap data.
Thank you. Dmitry Kosh. We can't hear you, Dmitry. Maybe we'll skip to the next one, and then you can just fiddle with the microphones. Okay, Jonas Mielke Möglich.
Yeah, hi, I'm Jonas Mielke Möglich, a PhD student from the Philipps University Marburg from Germany. And we are working on an automated moth trap based on the Aarhus trap from Kimberge. And last summer, we tested and compared the automated moth trap with conventional moth traps concerning moth abundance.
Exciting. Thank you.
So can you hear me now?
Yes, we can.
Okay. Okay. Sorry. So I'm a PhD student from the Friedrich University, Schiller University in Jena. And I'm working as a software developer for moth detection and moth categorization for the AMAT project. So I'm basically a software guy and would like to see what the hardware things are used nowadays and this whole speciality and so on. Thank you.
Great, thanks. Susan Hindinger.
Hello there. I am attending because I'm a leader at the of the nonprofit organization, the Vermont Center for Eco Studies in the US, where my colleague Kent McFarland is a collaborator on this project. He's the director of our Vermont Atlas of Life project, which is a biodiversity survey and monitoring project that also serves as a portal to receive and serve and interpret biodiversity data to inform conservation outcomes. And he's also a collaborator on the eButterfly project. So thank you for letting me sit in.
Great, thanks. Yeah, now I managed to mess up my system anyway, but I think I only have Alba and Tom on my list. So Alba, can you please introduce yourself?
Yeah, of course. So I'm Alba Gomez and I work at the UKCEH with Tom, Simon and David. And I'm a data scientist and I'm very interested in seeing how we can apply computer vision to insect monitoring and classification.
Great. Yeah, so Tom, do you want to say a few words about yourself?
Yeah, hi everyone. I'm Tom August from the UKCEH and I co-lead Easy Rider with David Roy. I guess I'm excited by everything in this project, but particularly how these tools can be applied in the real world at sort of national scale to support long-term monitoring of important groups like moths. Thank
you very much, Tom. And for myself, it just happens that I have the exact same motivation as Tom. So I'll just copy that. My name is Toke Høie and I'm a professor at Aarhus University. And we basically came into this from a monitoring perspective, biologist, and worked over the years with engineers like Kim Bjerre and others to advance the more automated ways of collecting data, primarily on insects, but also related organisms like plants and vertebrates. So yeah, without further ado, I mean, I was quite excited to hear this introduction. I know it takes a little bit of time, but somehow it kind of builds a nice picture of how big and diverse this group is and how well represented we are across at least parts of the world. Obviously, there are parts that are not quite as well represented, but let's keep this community growing. So please, everyone, pay attention to next invites for this series of workshops, or feel free to propose or even lead workshops like this. We by no means want to monopolize this group. It should really be to the greater benefit of advancing these tools and methods. So yeah, without speaking much more, I think we want to go through the presentations now. We have 6 presentations. And I've given the speakers 8 minutes each, so that there's a couple of minutes for questions. Then we'll have discussion afterwards, and we have the hope that we can break you out into small groups and have a little bit of dialogue there before we collect everything and finish up hopefully in an hour and 40 odd minutes. So yeah, without further ado, I'd like to introduce Kim Bjerre, my colleague here at Aarhus University, to give the first presentation. And I believe, Kim, that you can share your screen. So I'll give you a warning when 8 minutes have passed.
Okay, can you see a screen now?
Yes, we can.
Yeah. Okay, I would like to give you a quick overview of the automated moth trap we have developed here in Aarhus. We started in 2019 based on a demand from Toke that he would like to have something that could automatically detect the moths and try to make a small film of it. And I had some students that were going to work on a thesis on it. So I developed the trap and they tried to do some software in being able to track the moths and also classify them. And I think I will show you a small YouTube video on that. So here's what the trap is actually able to do. It's actually able to film live moths settling here on a white sheet. And the algorithm is doing that, is trying to classify, it finds the moth and then it tracks the moth. And then every moth are getting an ID with what species it is. So if we run the film, you will see that they're sitting still most of the time. Okay, here one moth appears and the algorithm tries to keep on tracking the moths while they're moving around here on the white sheet. So that's basically what the algorithm is able to do. And the whole system is to film the moths, observe them, and count the numbers and distinguish between the different species. And you can, of course, also correlate with the temperature and the position of where you're doing the monitoring. But the actually team of today was to dig into the hardware, so I'll not speak anymore about the software. The hardware is the newest design we have made here in 2022. We are going to set up 12 traps here in different places in Denmark. And it's based on different components that we have tried to find components that has a fair price and it's easy for most people just to build themselves if they have some experience on electronics. So you have a Raspberry Pi 4 here. That's connected with a USB 3 cable to a web camera from Logitech, and an SSD hard drive where you can store all the images on. And then we have built our own electronics that's supplied by 12 volts. It could be a net powered from the mains, or it could be a solar battery powered 12 volts. And we are also working on that part. I'll tell you a little about. And then the Raspberry Pi turns on when it starts to film the moths during the night. It turns on the UV light and the ring light, and then it just waits, the camera just waits for any movements in the field of view, and then it starts recording images each 2 seconds and store the data here inside the Raspberry Pi. So I'm not moving the data onto the hard drive before the next day when I hope recording during night is finished. We have a light table here, as you can see. So the first version, the idea was that the moths would be attracted to the light and then settle on the plate. But I have made some tests here for 2 years ago without the light table, and it seems to work also without the light table. The main thing to attract the moths is definitely the UV light. And the light ring here, the USB light ring, is the purpose is, of course, to get a good illumination of the moths when we are doing the filming of it. And here you have a picture of the electronics. So we have made a nice circuit board here with the electronics for the power turning on and off. And there's also a pot meter where you can adjust the ring light, the intensity of the ring light. And then the software is here on the SD card on the Raspberry Pi. That's running a standard motion software that you can download from the internet. So developing software on this part is really just a lot of configuration. And then, of course, you also need to adjust the camera, and you can adjust the mechanical distance to the plate for how near you will be to the moth when you do and you can also move it up and down. The solar power we have experimented with in 2020. We consume nearly 32 watts when it's turned on, and we are turning it on from 11 o'clock in the afternoon to 3 o'clock in the night. And during daytime, it uses that the Raspberry Pi itself actually consumes a lot of power, 5.4 watts. And we have tested the operation in 2020. And you can see we made some electronics that we can measure. The blue curve is the battery power, and the red curve is the solar panel that's trying to charging the battery. And you see here we have a battery that when it's turned down, the blue goes down, and then it suddenly drops to 8 watts, as it indicating that there's not too much power on the battery now. So we have tried to work with different configurations to see if it's working. So this is the optimal we have had. We have another system in Greenland that is also running on solar power and filming insects and plants the whole year. And there we have made a special battery control that can turn off the power to the Raspberry Pi during the time when it's not operating. By using that, we can actually reduce the power consumption from 250 watt per day to 130 watt. And that would, the whole idea is to use the setup where you have as a small solar panel as possible. As you can see here, we have made a lot of them. We have made 12 and actually bought components for 16. And we are going to set them up here in Denmark and record images in 2022 and 2023. We also made experiments because the problem is that when the moths are attracted to the trap, they will not only settle on the white sheet, they'll settle all around on the grass and in the nature around it. And many of the moths actually likes to find shadow near the light. So the idea was to build a setup where you have one in a height of 1.5 meter above ground, that then the probability of the moths only sitting on the white sheet should be greater. And we haven't tested yet. We have only made one prototype, and now we are going to test that also in the coming period. Yeah, let me see with the time.
8 minutes, please.
How many more long time do I have to?
Well, you spent your 8 minutes basically.
Okay, the last slide here is some questions to myself. Do we need the light table? Many of the mods are not only sitting, they are sitting outside the camera view. I think we can't do anything about that. As you can see, here's an example of the ring light is not the best. You have a centered light in the middle, but I know that some has used a squared light that gives a much more even light. Could we lower the power consumption, making, instead of using the UV light from Germany, build our own? If we're not using the light table, could we increase the camera's field of view, even seeing more species? And then, of course, the algorithm and the software, like I know that a lot of people are working on also. So that was my input.
Great, thank you very much, Kim. While we swap to Donald Hoburn, do we have a brief question? You can just speak out, I think.
Yes, I've got one question.
Yes, go ahead.
If I see it correctly, the screen is bigger than your camera, isn't it?
The screen, well, I can actually adjust here with the working distance from 7 to 26. If I adjust it to 23 centimeter, I can see the whole wide of the screen.
But your resolution is not okay. What we did is we made the screen the same size as the camera can have, so you don't have any insects around it.
Yeah, exactly. That's possible with this setup here. Yeah.
And what we also did is created a PCB with LED light, UV lights on it. I'm not sure if it's enough for you.
Yeah, I know that.
In my setup, I use maximum 5 watts even during the nights.
Okay, maximum 5 watt, because the UV light here, this one, use 15 watt.
Yeah, maybe it's way stronger, but that's something we can compare.
Yeah.
Great. I think we can have maybe a few more questions after the first 3 presentations that all deal with the same basic setup. So if you unshare your screen, Kim, then we'll move on to Donald.
Oops, sorry. That one.
Yeah, please go ahead, Donald.
Okay, thanks very much, everybody, and thanks to Toka and Tom and everyone for organizing this session. Hopefully my presentation will be slightly shorter because it really is just building on the same approaches. I've been interested for quite some time, as represented by this weird diagram at the top left, in thinking about ways that I could set about imaging, particularly night flying Lepidoptera as they come to light, instead of just relying on either manual capture or using a DSLR manually to identify them. And so when the paper came out in Sensors from Kim and the team with Toka, it was something that greatly inspired me. And so I set about here in Canberra building a close to exact copy of the design that Kim has just described, with the Raspberry Pi 4 and a USB 3 connection to the Logitech Brio web camera, a ring light, an LED light table, using the same motion detection software that Kim was discussing. And I took a decision fairly early on, because my experience of my 15-watt tubes was that the stability of the light produced over time was not great. I produced for myself a 9 LED tube of the same dimensions to replace it, just so I get slightly more consistent light. And so that one has 6 UV, 1 green, 1 blue, and 1 white light, and seems to function very effectively. As with Kim's discussion, the power of the Raspberry Pi 4 makes it possible to use motion detection software and therefore pause for long periods if there's no movement in front of the trap, but equally on a warm night collects massive numbers of images. I've been running an example of this in my front garden for probably 6-7 months now, and it works very stably. It collects images every night, and I've got the nightmare ahead of me of just trying to process all of those. However, I was even more interested in getting to the point where I had a setup that I could easily just carry into the local natural areas, leave out for 1 or 2 nights, and just get some data in different locations, moving around and comparing insects over time. There is so little time series data for Australian Lepidoptera that learning how to do this and potentially setting out 2 or 3 of these traps and just changing the batteries every day or 2 seemed to be an excellent solution. And so I decided to tinker with a Raspberry Pi Zero as a lower energy solution. It doesn't have the USB 3, so I shifted to using the Raspberry Pi HQ camera and the 16mm lens. And I tinkered around also with different numbers of LEDs, and I'm standardly using 6 at the moment, 4 UV, 1 blue and 1 green. And the only other light source is then the ring light. I'm using a 10-inch one rather than the 6-inch I started with, because it does give slightly better balance to the illumination over the surface. One of the features of the Raspberry Pi camera, and I'll show a few images in a moment, is that it has a different aspect ratio from the Logitech Brio. In fact, actually slightly more pixels, but it does mean that towards the corners, there's a lot more effective barreling of the image, so more distortion at the corners. And the other change was that I've written my own Python scripts for this one that turn on using exactly the same circuitry that Kim has described, but then follow a customizable time-lapse approach that typically I trigger on a cron job on the Raspberry Pi, but I've also set it so that I can manually turn it on and just leave it running. Because I was leaving this out in the field in different locations, and I am terrible at remembering to take coordinates, I added a GPS sensor to this. So first thing it does when it powers up is switch on the GPS sensor and wait until it's managed to get, I think, 15 readings from that sensor, and then uses those to derive a mean and some estimate of standard deviation to give an indication of just the accuracy. And I've got a temperature and humidity sensor from which I write those variables into the file names of all of the images as they're being collected, so that I've got ready access to that information later. This is a fairly typical image taken with the same setup as Toker and Kim have been discussing. The composition of the moth fauna here is very different. Massive numbers of ecophorid moths and tortoisids, and so the overall average size of the insects of interest is much lower, but most of these are highly recognizable from local experience. And so my challenge is that there are so many insects on the surfaces in this case that it takes quite a while if I want to go down to these smaller size classes in adding identifications. And of course, you can see that there's a lot of other insects there as well. This is an example with the light table set on relatively low illumination behind the sheet. And yeah, the image quality when you really zoom in is quite blurry, but even from this, those insects are all, certainly the moths, are all identifiable. And you can see caddisflies and small cicadelidae and things like that on the sheet. This is the result I've been getting from the Raspberry Pi camera. So far, I've found that controlling the exposure is quite difficult, even if you turn off apparently the automatic compensations there, it does tend to become wildly overexposed if anything large and black lands anywhere in the frame. But again, the image quality is fully adequate for identifying the insects under study. Most of my attention now, and I must admit that over the last couple of months I've been diverted onto other things, is around looking at how I can do some manual processing initially of the images that have been collected to produce data and metadata packages that could flow on into the Atlas of Living Australia, GBIF, and other destinations. So what you can see here is mostly automatic. Okay, I'm just finishing. Mostly automatically captured metadata, including coordinates, as you can see. Summary that is produced of the captured images, and then using Kim's code with a couple of further modifications, some processing to derive blobs, and at the bottom, a manual tool I'm using for associating identifications with those, the sets of blobs that form a track. And the intention then is hopefully to get to enough identified images of most of the common species that I can start experimenting some more with machine learning approaches. Thank you.
Great, thanks very much, Donald. This is all very exciting. A question for Donald before we move on. Yes, Jörg, just go ahead.
Yes, thank you for seeing me. I have a general question. How do you, at least in my experience, the moths, they go to the whiteboard and then they may fly off and come back. So how do you avoid double counting?
I'm sure that there is plenty of double counting going on. My expectation was ultimately that I would treat my measure as being the maximum number of individuals of the same species recorded at any simultaneous point in the night, which often rises quite high in the context I have here. Additionally, the number of frames in which the species occurs, but you're quite correct, especially on a warm night, the amount of movement makes recognition of the same insect genuinely pretty much impossible. So my experience is rather different from Kim's in Denmark. Most of my insects don't sit still very long. Some of the larger geometrids will flatten themselves out and wait, but yeah, you could see there were mantis flies scuttling around trying to catch things. We get large ants running all over the place like sheepdogs chasing all the insects. So it doesn't settle down for long on a warm night.
Okay, thanks very much. I think we'll have to move on to keep the time reasonably well. Simon Teagle, are you ready for sharing your screen?
Yeah, right. Can everyone hear me?
Yes.
Perfect. Let me just put this in the presentation. Okay, so hi, yeah, Simon Teagle. We are replicating the Art House design to make it more widely accessible. What that means is we took delivery of 2 of the Art House designs at the back end of 2 years ago, 2020, and we deployed them for 6 months, 8 months in some sites across the UK, where we were able to learn about what they do, how they work, and count various biodiversity. So for those that do know, I apologise if I'm going over something they've already heard. They were deployed with what was called the Network Rail project, which was a biodiversity study of line-side UK biodiversity counting. We deployed them alongside small mammal cameras, bat microphones, bird microphones, and various weather sensors as well to capture line-side activity. From that, the lessons we learned using the moth trap received from Aarhus were around how we would not redesign it because it worked really well, but how we would replicate and build on that design to make it more viable to the wider community. So we worked with physical parameters like size, weight, number of components that it consists of. As Kim showed, a lot of their design comes with variability in how high and distance of the camera from the target. features like that, we considered removing, having that variability away to make it easier to machine, easier to reproduce, and easier to replicate. We learned about the power consumption of the moth trap, as the power consumption showed from Kim's graphs. It is a power hungry tool, that UV light, but we learned what kind of power capabilities we could do with it. We settled on a 120 amp hour battery and a 100 watt solar panel. I can go into more details of that if people need another time. We learned about the image capture sensitivity, as Donald and Kim have both mentioned. It's a massive amount of data, a huge amount of data. So how can we play around with the threshold for the motion detection, the blob detection, and either reduce that to a point where we're still all happy within the community that it's still capturing enough images, but we're not hoarding mass amounts of data that's duplicated effectively. Image quality, everyone's touched on this already, but how we could optimize the camera settings with a larger depth of field and a wider field of view. That, I think, was more user error on our side. We could have done a bit more work with the camera in our labs before deploying it in the field, but nevertheless, it was a good lesson learned. And we worked around system robustness. So we had a few issues with the SD card, loading everything to the SD card and that being downloaded, if you will, to the SSD once every day. So we changed a few processes on that to bring things more robust and have less chance of corrupting our SSD at a later time. So with all that in mind, we simplified our design, which now looked like this in prototypes. We had a single base plate. We were able to modulate things like the camera and the light box, the UV lamp itself, the backboard, and the control electronics. So anything, if we delivered these around the world, anything that needed to be changed could be changed in modular format as opposed to finicky on-site work. We removed the light table. We tested the same as Kim mentioned, the effectiveness of the moth trap without having a backlit light table, and found it to have very little to no effect on the activity of the moths. So we've removed that and removed the PCB as a result of that. We've reduced the electronics because there's no light table, and we've improved the robustness in the system by adding a watchdog to the electronics and changing how the data is stored. So the data is stored directly to the SSD for us. The SD card only holds the program for the system. We would have everything on the SSD, but to enable removability of the SSD, so data access on an easy basis, it's easier to have the program running on the SD and all the data on the SSD. I'm probably losing you all, so I'll move on. We installed a watchdog, which is what I said, which basically fail-safes the system if we lose power. It's a program that, well, I'll show you in the next picture. And then image quality enhancements. We spent a bit more time in the lab working on optimizing the distance we have from our camera to our board, which we set on a set distance. It's not on there, but I think 240 mil. We've got a depth of field of about 30-35 millimeters now, where everything's gorgeously in focus. So if we want to play around with different surfaces on the backboard, like materials or foams or impregnated surfaces to attract and lure the moths, we've got a good depth to play with to stick anything to it and edit it as we go. Okay, so as I said, it's now modular. This is just an image to show with the sections that are all separate from each other, and then they all collapse and screw together. So separate UV light and holder, separate electronics box, separate backboard, and a separate base plate and frame. Our electronics looks like this. It's like a small compact box. With reference, this is the kind of size of it. So it's not much bigger than my hand. Looking in it on the top, you'll see we've got what's called a watchdog. That runs a program that counts down, runs a countdown every now and then. And when the countdown sees that the trap is receiving no power, it sets the Pi to turn off, stops the motion software, and shuts the Pi down safely. So there's no risk of the SSD being corrupt and losing that day or night or weeks worth of data. Other thing it does, which I think is quite a nice touch that we didn't know it did, was if the Pi hangs for any reason or the program freezes, that will also kick in this countdown timer, and in turn it will reset the Pi. So If there's an error that occurs during the night, we'll catch that, correct it in a reset, and restart the system. And we've got our Pi there with the step down for the 12 volt coming into it. We've got the SSD controller. This is a 12 volt timer relay. So this is how we compute and control the monitor and set it up and then leave it and come back in 6 months' time. We can set a timer on that to come on every night, every other night, every once a week for however many hours across that night. So that sets your program regime, if you will. And then the rest is the wiring for the Pi, the SSD, and the power.
Time is sort of up, but
okay, I can flip through, don't worry. This is picturing what the work that's been going on, showing the depth of field we've got with the hand and the background picture we're using to set the focus and the depth of field of the camera. And then the last screen is a few questions really I was going to pose to the science community across the next deployment period of the summer. So it'd be good to have feedback on the best and most effective backboard materials or lures for animals to sit and stay, let's say, or insects to sit and stay on the backboard. The lowest threshold we can have of the motion detector and the smallest image size possible that would still be usable as a classifier to save on data, volume. And the bottom right image here is an image we got of a crow that was creeping in to try and sneak some of the moths for dinner. It'd be quite good to have like a little less important competition if we can get the best image of a non-insect. Happy to take questions.
Great. Thanks a lot, Simon. This is all very exciting. Questions for Simon? Donald?
Yeah, just a quick one. So your timer switches off the whole thing except for the few hours a night when it's running. Is that correct?
Yeah. And it runs on a set clock then for that after. So when that's powered off the battery and when that program's set to start again, it powers the whole thing back up.
Yeah, okay, good.
Joe?
Hi, I guess a bit of a general question for anyone using these systems and whether you have them just on the ground, because here in Newfoundland, I often refer to us as the New Zealand or Australia of Canada, where we've got many introduced species, and many of those small insectivores, toads and shrews and things that are extremely dense in the summer. Like I'm talking difficult to walk through the forest without stepping on one of them at certain times of the year. So that tends to be a problem. Even the toads in some of the light trapping areas that I've had, light walls and things, are managing to find ways to hop onto logs and hop up onto these platforms to get at moths. And so it's been a real challenge for us. I'm just wondering how you, do you often put them up high, or anyone else having issues like that?
I can just jump in on our one. We've built 4 small legs that come out the bottom of our base plate, and they're what's mounted to the battery box at the moment. So it will sit on the mains, or the battery, the 12 volt battery box, which is about 1/2 meter off the floor. To have that any higher, we can build leg extensions, but they're tapped poles basically, which, to be fair, you could probably use scaffolding to slot our current legs into and extend it yourself on site. It would be good to get feedback on an ideal height across the community, like a standardized height, if you will.
Yeah, I agree. This is a discussion point we're also hoping to have in the breakout rooms as well. But good questions and good answers. Jörg, you have a question too?
Yes, thank you. What is the cost to build such a trap?
For UKCEH, these systems we've made are £2,900.
Okay, thank you.
I thought I'd just quickly show this as well. This is a machine cutout of the camera and the light box. So we're using a rectangular LED frame, which will sit into that sunken recessed slot, and then the camera slots directly into there.
Looks great. Maybe both Simon and Kim and Donald can type in a few cost estimates in the chat, so we have that for everyone. Okay, we'll move on to next speaker, which is Gunnar Brehm, who speaks about the Amot moth trap design. Go ahead, Gunnar.
Yeah. Let's well, wait a minute. Bildschirm frei here. Yeah. Still trying to figure out. Yeah. Okay. Can you see it?
Yes, it's fine.
Okay.
Thanks.
Yeah. Yeah. I'm talking now about a different approach of a camera trap, which we call the moth scanner. And it surprisingly comes out with a very similar result as you have seen in the Aarhus trap design. So this is basically what we have been using since 2 years. There's a light, UV light with LEDs using approximately 4 watt. And we have installed a screen with a size of 24 by 36 centimeters. And we're using a 20 megapixel camera with a rather good resolution. I think a better resolution than the Raspberry or the Logitech cameras. We started that in 2020, and I say that because we have not been aware of the Aarhus trap project, but the results or the design is really surprisingly similar. So we have this screen which has a similar size and a similar position, and in front of it the camera, on top of it the light. And I'm going through a couple of points that we checked for until we came to this design, or which we are currently working on. And one is, of course, UV is the most attractive wavelength. So we need UV for attracting moths or other insects. I don't think other wavelengths are necessary. Or in some cases, you only want to use UV because then the trap is also not very well visible. So this is one example image that we took last year. So sometimes we had really a full screen, many different kinds of moth, species of moths, some common, some not so common. And this is an example how the resolution of those images is. And we think if you really want to identify or run artificial intelligence, especially on the small species, you need to have a resolution like that. So we considered also using either a larger screen or a larger sensor, but we think it's a good compromise to have this sensor size and this screen size. When I first started, I thought it was a good idea to have use some kind of box that used to have edges like this. But the problem about a box and edges is that many of the insects were trying to hide somewhere in the corners. And then there were also reflections. So that was not a good idea. So I skipped this rather quickly. And we came to the design we're now using. Now, the position of the light. I tried out several designs, so you can put the light underneath the screen. That basically worked. It was okay. I also came to the conclusion that the screen or the table itself does not have to be illuminated. It's really something different. You need one lamp that attracts the insects, and then you need a place where they sit. But the place where they sit does not have to be illuminated. We thought indirect illumination with UV could be a good idea, but in fact it was not a good idea. It did not really work well. So direct light works better. And we also tried what I thought was very promising, to integrate the lamp into the table. So you see a little bit of violet here in the center of this table, but that also did not work really well. And one of the problems about such a transmission model is that so much radiation is then absorbed by the materials. It really makes more sense to have it like this. And so we chose the position above.
We didn't care so much about rain protection here because we could use last year this small hut in an abandoned garden. So that was fine, but it worked well. And I also, I'm thinking about alternative positions of the whole camera setup and the screen. So this is basically the design we have been using so far. But the problem is if you use such a horizontal design, there could be better alternatives that are easier to protect. And there are no individuals sitting on the backside or somewhere else. So I considered turning it by 90 degrees and taking photos from above. So this is a very new idea. So far, I have only tried it out in the lab with one species, Spodoptera. And in this species, it turned out that this design does not work at all. So the moths don't like to sit on this flat table. They rather like to sit on a vertical structure. So the first tests really showed this is really working better. But it also came to my mind we might try it by rotating it by another 90 degrees and taking photos from below. That actually might work because moths might like to sit on the substrate like that, and I want to find that out later this year. We think that using a scale bar is probably a good idea. We haven't included so far in our approaches, but we think it's always a good option. And I apologize that I've not been able to send around the scale bars so far because they are still in production, and I hope we'll get them in the next 2 weeks and then I can send them around. Now, 7th point is light quality. So the light we're using to take photos. I don't think it will be a big issue. Even if you use a light of a bad quality, it probably doesn't affect so much the quality of identifications. But one should be aware that the coloring rendering index is ideal in daylight and also nearly ideal in flashlight. And there are very good LEDs, but there are also some very bad LEDs which don't have really good coverage of red and violet. So that could turn out being a problem if you compare data sets later. So I encourage users to check what the quality of their LEDs is they're using. So better quality is certainly a good idea. And then another point is insect or moth behavior. We observed that some moths rest more than others, so this will bias the results. And I think one of the next workshops will deal with this problem, whereas others are more active and rarely sit. We can see that in this image. There are a few Ilemas, they are sitting all the time, they're not moving much, and then there comes a knock to it. It shows up quickly and vanishes quickly. So you don't have that very well represented. And I think it makes a difference to traditional traps, light traps and light trapping. Ilema, I already mentioned that they tend to sit there. Some geometrids sit rather long in certain spots, but you have relatively few observations of hawk moths and noctuid moths and other groups which only come quickly and go again. And so, yeah, that was my presentation. Thanks a lot for your attention.
Great, thank you very much, Gunnar. This is really nice to see and nice complementary issues you're bringing up. Quick question to Gunnar before we move on. I mean, in the interest of time, then I think we'll just move on, and you can obviously ask questions directly to the speakers later if you think, yeah, if you care to. So next speaker is Jeremy Gulnik, probably pronouncing this a little bit wrong, from France. Please go ahead, Jeremy.
Yeah, thank you. So I'm going to present a project which is called Lipinoc, and we aim to develop a scientific, a participatory scientific program to monitor moths using smartphones. So Lipinoc, in a nutshell, we have two main objectives. The first one is to improve the state of knowledge on moths in France, because there is no organized monitoring network as there may be in other countries like in UK, but also to raise awareness of nocturnal biodiversity protection issue, because in France, most people are not at all aware of the importance of nocturnal biodiversity. So our participatory science program is for managers and for green and natural areas, but we also want to put it in place with general public in the next step, and in order to monitor a private garden. And we are supported by various experts from ecology, from EI, and a researcher from the French National Museum of Natural History is also the scientific reference of the project. So the device and the protocol we developed were developed in order to answer to two main questions with two different temporalities. At a short-term scale, we want to study the influence of anthropogenic pressures on the structure of natural Lepidoptera communities. And on a long-term scale, we would like to study the variation in abundance and distribution of nocturnal Lepidoptera species. But we will only focus on macromoths and potentially on a very distinctive macromoth due to image quality issue. We will also focus on open habitats, and our project have three main temporalities. Last year, we put in place a proof of concept. Now and for the next year, we'll improve our device and our protocol with the lessons from the POC, and we will develop large-scale animation tools for the national launch. Okay, so for the device, our device is based on a smartphone, and in fact we have a lamp, which is a waterproof lamp of a power of around 10 watts, which is only turned on at nightfall thanks to a twilight sensor. And then once attracted, the moth lands on a white felt, which is in fact stuck on plexiglass plates, and rugged outdoor smartphone with a 13 megapixel camera take pictures every 15 minutes. The device, the frame of the device is made of aluminum tubes, and the device is fixed in the ground with a ground screw post. And the lamp height is 1 meter. We have a light duration of around 8 hours continuously, and the price of our device is 350 euros for components only, and around 400 euros including manufacturing by FabLab. Here you have some example of pictures, and as you can see, pictures are darker on the side and on the bottom of the picture, and the lamp generates a backlight on the top where moths are not identifiable at all. And as you can see, the quality of the picture are less good than other projects. However, we found that identification percentage was still good, and these results are from all sites, all sampling device, and all sampling session, and we do not make any distinction between macro and micro moth. But as you can see, for around 10,000 moths which were taken in photo, we had around 60% of moths identified at a family level by an expert, 40% at the genus level, and 20% at the species level. Regarding existing challenges, we had some issue with the ease of use for participants, because it was complicated to assemble the device, which greatly impact the quality of picture and user experience. Furthermore, the device was too heavy, which was a shortage for participants, because it was sometimes difficult for them to put in place the device. And the smartphone application must be improved, and it's also impacted the quality of pictures and user experience. Furthermore, the device is too expensive for mass adoption, and we had also two main issues, which was an unsatisfactory scale bearer system, but also a poor idea of the distance of traction of the. Concerning next steps, we are developing a new version of the device, and our goal is to reach a device that weighs around 5 kilograms, which will be very easy to use, with very little variation in the position of the smartphone or the feet. We will reach a cost of 350 euros, including manufacturing and delivery. And we want also to increase the picture quality by reducing the distance between FEP and smartphone. And our first result here, again, without making distinction between macro and macro moth, show that we found around 80% of identification to family level, 60% to genus, and 50% to species. We are also developing a mobile application and a web platform, but we have still some challenges regarding scale bear and distance of attraction. Thanks for your attention.
Thank you very much. Questions to Jeremy?
Yeah, I've got a question.
Yeah, go ahead.
So that's great. I actually think trying to figure out a really low cost mechanism for citizen science is an excellent idea. I'm just wondering if you can see a way to lower costs down below what you have at 350 euros. It's still pretty pricey. I know like mass producing or producing at larger scale would lower, but like are there things that you can do to reduce the cost further?
Actually, I think if we work without an industry, it's impossible, because we would try to reduce as much as possible the price using a smartphone. We didn't find any way to decrease the price.
Yep.
Great. Well, let's move on to the last presentation then. Jacob and Rob.
Jacob, are you there?
Yes,
right.
Just started sharing it.
Looks good.
Okay, wonderful. So I'm Jacob Idek. I'm a first-year grad student with Rob Grolnick at the University of Florida. Rob and I are pretty new to the sphere of creating automated insect light traps, but we've done some kind of thinking and loose prototyping around how we might create cheap traps to deploy more widely. So this will be a pretty short presentation with more ideas than sort of fully engineered and tested solutions, I would say. So bear with us. We're also especially interested in building out a network for standardizing and distributing data. So we'll be writing some small thoughts on that toward the end as well. So we're largely interested in questions of insect abundance and phenology across the US, especially change in abundances over time due to human activity and how those might vary spatially. Also how activity regimes may differ or partition by species. So our goal here is to create a network of automated light traps in the US. And to sort of move towards this goal, we worked on some recipes for cheap traps for either us to build out and send to people, or for others to make using our simple kind of recipes. Yep. So this is our solar-powered pie trap that we produced.
I don't think your slides are advancing.
I forgot to. That was the wrong. That was on my other monitor. Sorry about that. Okay, so this is the that's the intro. Here is the field solar powered pie trap. So we put this together a few months back, trying to create sort of the cheapest solution we could put together for a solar powered trap that we might have in the field for a reasonable amount of time and be powered over time. We got it down to around $500 to $600, and it looks something like this. We're using a ring light to illuminate. The electronics consist of a Pi Zero and a Pi high quality camera, similar to Donald's model, for example, and also the same kind of ring light that others have used as well. Trying to get cost even cheaper. This is something that you might plug in your backyard. We're just essentially putting the electronics module into a plastic container. This gets things down to about $120. But then again, you sort of need to be near a source of power, for example, a campsite or a backyard for this to function. The getting it done cheaper still is sort of a phone-based solution. So this is sort of more of a citizen science kind of approach, where people could put these together very, very cheaply, probably around $40 to $50. And it's also especially easy to set up. This is something that people could build themselves. We use this free PhotoTrap Android app to take pictures every X seconds. That gets uploaded automatically to Google Drive. Like I said, it's very cheap. There's also a real possibility of creating sort of a specific app that citizen scientists could easily access and use to sort of easily specify metadata and very easily upload and share data as well. So our next steps for these sketches is to test them more specifically in Florida, both in backyards and in the field. We plan to 3D print or fabricate better boxes, build out some solar kits to send to colleagues based on that first field solar model. We also want to start planning how we're going to interface with existing processing pipelines. And really start trying to grow a network in the US. There's strong demand from the research community, but not much know-how in terms of how these things might function and be deployed. Finally, some rough ideas on data. We know it should be standardized with rich metadata, including some of these things listed. Ideally, it should be in a central repository where citizen scientists can easily upload it. And researchers can easily download it, including both raw data and process data. And it should be in a format appropriate for a wide range of downstream processing and analysis. Lastly, some open questions we've been thinking about are how process data should be handled and connected to pre-processed data. For example, I think it's clear that like every researcher might not want to run a complex ML pipeline themselves. They might just want to access occurrence data, for example, for a location. So how should that be connected to raw data? And to what extent should intermediates be saved, for example, coordinates, bounding boxes, or segments of moths per image per time step? And that's really all we'll be talking about. Thank you for the opportunity to talk through some of these ideas, and feel free to ask any questions about any of this. Thanks.
Thanks a lot, Jacob, and thanks for making these putting these questions out there as well. Great. Any questions for Jacob or Rob? Well, I guess we can talk a little bit about this in the breakout groups as well. But one comment I might have is that it's maybe worth considering price as some end of a continuum of price versus quality. Even in a citizen science context, I could imagine that the the reward for the users involved is also in the quality of the data that you can generate from these systems. So some users might actually be willing to pay a slightly higher cost at the benefit of maybe having a higher taxonomic resolution of the data coming out of the traps. So maybe it's still a discussion point whether you want to make these as cheap as possible, or like finding the right sweet spot between image quality and hardware design.
Yeah, I think you're absolutely right. Like there'll probably be a range of sort of what people are happy to shell out for these kinds of devices, for sure.
Yeah.
Hey, Toke, I wanted to just jump in and say I agree wholeheartedly, but I think it's always going to be not just about image quality, but about downstream applications. So like, what is the end purpose of the set of uses? And I guess like the sweet spot might actually be more about the minimal image quality that provides the maximum amount of downstream utility broadly, right? So it could be some kind of trade-off that also includes like downstream value, right? Utility in many, many different kinds of questions that can be asked.
Yeah, absolutely. And I guess we are still only just beginning to quantify those trade-offs. So let that be a kind of segue into the discussion part of this this workshop. We are actually planning to put you into breakout rooms. Alba is kindly offered to organize this breakout room discussion. I just want to say that we've tried to organize it so that there are members of the EasyWriter team in the breakout rooms that can maybe provide context for some of the discussion. Feel free to discuss whatever you want in these breakout rooms, related obviously to the theme of the workshop and the presentations. I did put a few discussion points in the invitation that you might have seen that relates either to how we attract insects and what's kind of the bait element of these traps. The image quality, I think we've got a nice kind of variety of approaches to hit these trade-offs between image quality and trap costs. And a third discussion point we could propose you talk about is the scalability of these systems. How do we move forward to create global standards for insect monitoring that I think would be to the benefit of the research community on one side, but also those that can help actually look after our biodiversity more broadly, that we have standardized data in the future. So those were just like inspirational points to this 15-minute breakout room exercise we'll have in a minute, and then we'll reconvene after those 15 minutes and try to summarize the main points from the discussion groups. And I would kindly ask the members of the EasyWriter team to take some notes. So if there are points that will not be reported back before we have to close this at 35 minutes from now, that we at least pick them up as a team and can bring them forward to the community more broadly afterwards. So yeah, before we do the breakout session, are there any other general comments or things that you would like to add to the discussion elements for the breakout room? I think we can just hear quickly if there's something you want to add to the discussion points for the breakout rooms.
I think in terms of the just adding to the scalability topic, I think sending data remotely. So I think a lot of the solutions currently seem to rely heavily on going into the field and getting the data one way or another. So yeah, it'd be nice to talk a little bit about ways of uploading the data automatically to some data portal or whatever.
Great, good point, Julian. Anything else?
Toke?
Yeah. Neil? Yeah.
So I don't want to introduce too much mission creep in here, but I also think in terms of scalability, how does this model work with other situations? So I'm actually probably more interested in pollinators, but I see this basic trap as being very, very useful for modifying it for for pollinators. But I would, in the back of my mind, think about other things like ground dwelling arthropods, blah, 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 blah. And then also to think about just completely different habitats. So when we talk about the height of where this should be, if you're in the tropics, you're going to probably want to put these throughout the canopy. And so I think we should think about all these different things in talking about these.
Absolutely, Neil. And if we don't have enough time now, we should organize a separate workshop that kind of explores the applications beyond nocturnal insects. A lot of us are interested in that too. So yeah, do take that point forward in the breakout rooms. Right, let's get going on that, and we can kind of summarize towards the end. I would leave that up to Alba now to move us into the breakout rooms. You okay with that, Alba? Great. See you in 15 minutes then. So which one do I join, Alba?
To the number 6. I cannot find your name, but number 6. Yeah.
Okay, good. See you in a bit.
See you in a bit.
This meeting is being recorded.
Sorry, Jacob.
Oh, it's all good. Yeah, but color standard, scale bars.
Yeah, sure.
Hello again. Welcome back. We got cut out in the middle of a sentence. I don't know if that happened to any of you other groups.
Yes.
I guess that's a good sign, isn't it? That you weren't bored or you didn't know what to talk about. So I found this extremely stimulating, and I hope you did too. It's really great to have these kinds of conversations. We still got 15 minutes. I'd like to keep to the original time, and I think it would be nice to have a brief round of reporting from the various breakout groups. So if we can have a representative from breakout group 1, if you know who you are, to say just like a few main points from your discussions.
There is no room 1, it should be room 2, which is
sorry.
Tom's. Yeah, sorry. Yeah.
Tom's group. Okay, room 2.
Yeah, great, thanks. So I had one of the Davids, Michael, Jeremy, Daniel, and myself in my room, and we actually spent most of our time talking about translating to pollinator monitoring. We all had an interest in that. We talked about having the trap height at the height of flowers. We talked about needing to have attractants that would attract the varying types of pollinators. So we spent quite a bit of time talking about the color of the background sheet, and is there an optimal color, or are we going to be needing to combine different colors to make sure we attract the range of insects that are attracted to different sorts of colors? We talked about combining multiple cameras together on top of one set of hardware infrastructure. So just having one solar panel, one battery, but perhaps 2 or more cameras looking at different colored panels, or perhaps a moth camera and a pollinator camera combined. These sorts of opportunities. We were curious to learn more from Diopsis about how come they settle on the yellow background for pollinators, and how well that worked. And we talked a little bit about pheromones, but thought that might be perhaps a bit of a red herring. We then spent a little bit of time at the end talking about smartphones and sort of the low cost end of that sort of continuum. We're all quite excited about what's possible. All agreed that something that can engage the public at large in getting excited about and monitoring moths and other insects would be great. But sort of concluded that there's going to be we need to think about that whole spectrum. Some people will need the professional grade, which you can stick out and in Greenland for over winter, and it will turn itself on when the snow melts, and it will run all the way through the summer. And also at the other end, where sort of my mum can get something for Christmas that she can use, and all the way in between as well. So we need to keep our eyes on that whole spectrum of different tools that will work for different projects.
Well, great, Tom. Thanks. And you figured out a way to standardize all that data into one big.
Yeah, it's in my notepad just here. I'll email it around to you.
Great. Yeah, absolutely. Great. That's very nice. Any additions from that group?
Perfect summary.
Yeah, great. Okay, let's hear from breakout group 3 then.
I might be the one to try to do this, guys. Joe's nodding emphatically that I should try to summarize our conversation. Great. So our group was Jamie, Donald, Joe, and Kim. I'll note that one of the unique parts of our breakout was that we often lost Donald midstream in them, and right when he had something absolutely brilliant to say, we'd lose his Wi-Fi. So poor Donald, I think that's why he's not the one doing the summary actually. So we covered more about image quality trade-offs to start, and really thinking about how those impact our ability to build, on the machine learning side and the processing side. And so a lot of our conversations kind of moved from trap design to the challenges around classification. And one of the things that I thought was really fascinating about this conversation, I'll just summarize the sort of the 2 or 3 really key kind of innovative points that I hadn't thought hard about before. But one was, basically thinking about how we can use these systems to detect new things that we don't expect. And so Joe mentioned that, there's a real need from the agency side up in Canada to be able to detect, moths that might be exotic or invasive, and how hard these systems are going to be pressed to basically do that on the machine learning side, given that classifiers aren't really well adapted to sort of discovering novelty. In fact, they're not adapted for that at all. And how much we can leverage the power of having sort of more systematic collections of data across broader regions and scales to sort of fill the gap, so that we can actually be able to start getting towards that kind of discovery. And I think it was Jamie who mentioned that, that would also lead to sort of new innovations in machine learning, where we could really use information about the tree of life and uncertainties as we move from the tips down to the, or from the deeper parts of the tree up to the tips, to make better assessments about how certain we can be, say, the species level versus a generic assessment versus a family level assessment. And I think that's effectively the time we had and what we mostly covered, but I'll turn it over to the rest of the group for anything I missed on that. Go for it, Joe or Jamie or Kim or Donald.
Yeah, it looks like the group seems to be happy with your summary. Great points you're raising there. I think we'll just collect all of this and then we'll have a bit of discussion towards the end if time permits. Thanks, Rob. So I guess we're at group 4 then.
Yeah, I'll take it for our group if you guys are happy for me to. Part of my pronunciations, we had Jörg, Gunnar, Jacob, and Laura in our group. We started off just discussing a bit from Laura's point of view, how beneficial it is to see all the different designs and the way that she can try and build a design for herself for her own project to get into the field this year in her general project. And then without disappearing too far down the rabbit hole, we started to talk standardization with regards to initially hardware, well, initially actually metrics, metadata, but then moving into hardware, and how, I guess it posed the question of, do we want to start looking at standardization from point of use this type of camera or this camera and build it at this height and be this distance from this set size of sheet? Or is it more a case of looking at the whole group as a whole and saying, can we not use baselines instead of standardization and say, actually, we need a camera of at least this quality, being a pixel size or yields a pixel size of at least this size to get this higher or X amount of quality from an image to using a classifier. So creating baselines that give a flexibility for the design and budget, as opposed to being so strict on the standardization in itself. And then we ran out of time. Anyone else have anything else to add?
Great, thanks, Simon.
My pleasure.
It looks like you covered it well there. Okay, group 5.
I think we were group 5, weren't we, Alba? So I'm happy to have a go, and then others can chip in. So we had Maximilian, Neil, Jonas, and Luca, I think. I didn't take notes at the time. I think remembering who was there. So yeah, I guess, you know, going last in these feedback, you're always reflecting and repeating what other people have said. So we covered some similar ground. So quite a bit of a discussion around different use cases for the systems, you know, the extremes of putting it out in the tropics and leaving it for months versus having it in a local reserve where you're visiting quite often. And then discussing what implications that has for things such as what processing you might need to have on the system itself, rather than necessarily storing everything, given the constraints of storage and particularly power. You know, writing to disk can be power hungry, and issues like that, and issues that to do with, you know, the temperature range that things need to operate in needs to be considered. And also in terms of use cases, what your target organisms might be, you may alter how you deploy the traps in terms of heights. Different insect groups may be operating at different heights in the canopy or vegetation. So I think we sort of came round to the sort of view that we really need a very adaptable system to cover a whole spectrum of use cases, and then to be highly configurable. So some standardization in the sort of the technology, but almost pick and choose what you want out of a set of options to design your own trap for your own use case, environment it's operating in, and target organisms, for example. So that was, yeah, we had a really good discussion around that, and learning experiences of those in the group who'd run systems in different contexts. And then we also had a discussion around the main audiences and whether we're sort of aiming for a research grade, highly robust system that's highly repeatable, that may be used by, you know, big organizations just deployed at multiple locations, or whether but also at the other end there is a citizen science requirement, more do it yourself, cheaper options, and we can definitely see value in both ends of that, and again, another spectrum of use. And we thought we'd be interested to know what the sort of citizen science audience is, what they're asking for, which may direct how those things are developed. But we welcomed all the innovation in this space, because I think there'll be lots of ideas emerging across this excellent community here and the wider community, and we expect, you know, some surprises to come out in the next few years in this area. So yeah, really great discussion. Thanks. And anybody to add anything that I've missed?
Thanks, David. Any additions from the group? Great. Okay, I think, yeah, I can represent group 6. We were Dimitri and where are you all now? Julian and Mark. Yeah, great. So I think we had a slightly different discussion. I think we started out talking about the pros and cons of storing the data locally rather than submitting it over network connections, 4G, 5G, or Wi-Fi, and the motivations to do either. So obviously, it is in the interest of some communities to minimize the cost of operating these cameras, and if the system itself can submit the data to an online repository. You don't need to go in the field to collect the data, so that could be a benefit. If you're recording in a remote location, maybe that is a trade-off against the power availability to submit data over any wireless connection. So we talked a little bit about that and reflected on other potential reasons for having this completely autonomous, in the sense that you could get the derived biological data in real time. And I guess we touched briefly on the opportunities for integrated pest management. Maybe that's where you need real-time data more than in the biodiversity surveys. But clearly those kind of hooks are something we need to keep in mind as we continue to raise funding for these tools. So if there's a particular reason for having a real-time application, then obviously we need to move forward building the solutions to that. I guess we talked a fair bit about image quality and this trade-off between quality of the images versus the cost of the system, mostly agreeing that the higher the image quality, the greater the opportunities are. So at this stage where we are still basically grappling at the kind of beginning of this adventure, I think most of us agree that the higher the image quality, the better, the more opportunities we have for the future. But that shouldn't lead us to forget the scalability issues of this in the longer term. We talked a little bit about how these high-end cameras work in the longer run, how many images can they actually take, and are digital SLRs really suitable for taking hundreds of thousands of images versus compact cameras versus the Pi cameras and so forth. So there might be something that is worth thinking about there too. And then I think we ran out of time. Dimitri was saying something clever towards the end. Maybe you can now finish that sentence.
Yeah, we are talking about capturing even more images, not like every minute, but every second perhaps. And I thought of not capturing images anymore, but perhaps small videos. So you can use video encoding to reduce the number of or the storage you need for that. That was the final thought.
Absolutely. Yeah, I guess this was when we were talking about the downstream data that we're really interested in collecting, whether this requires tracking individuals and correcting for the bias and how many times we get records of the same individual, or whether we want any behavioral information out of the traps, or just want like time series subsampling of the community that we are recording. Great. I don't think there was a group 7, right, Alba? No group 7. So unless, yeah, anyone else have aspects that were discussed that weren't quite reported back? Not really. Okay, well, time is almost up. I think we could continue this conversation for quite a while, but since we're only 1 minute away from the deadline, I just want to reiterate that we have 2 other workshops lined up. One is emphasizing the challenges and benefits of integrating these new tools into existing monitoring activities. How do we compare the data or make sure that these new tools will collect data that is somehow comparable to the ways that such data were collected traditionally. And we have another one further into the spring lined up for the details of image analysis, more on the machine learning side of this in these initiatives. I think we need to have more than these 2 workshops, and any ideas you might have for follow-up workshops, be that applications for other taxa or other domains. I think that would be very interesting too. And I think the Easy Rider project will continue for a little while. We will have other projects that will also continue these conversations. There are people here from EuropaBon and other big initiatives. So yeah, get in touch and we'll make sure that we reach this community if you want to have follow-up workshops like this. Yeah, with that, I really just want to thank you all for showing up and being such a active group for the discussion. So that's greatly appreciated.
Thank you, Toke, for organising
lot.
this session. It's been really enjoyable.
Yeah.
Thanks very much indeed.
Thank you.
Thank you. Bye, everyone.
Thank you very much.
Bye.
Thank
Bye.
you.
Thank you.
Thank
Bye
you.
bye.